First, I wanted to introduce Alice Wu, who is a senior associate in science policy working on climate and meta-science policy for the Federation of American Scientists. She will share more about how Metaculus and FAS are collaborating and how this collaboration relates to today's talks. Alice, you can take it away.
Awesome. Thanks, Christian. Yeah, we're very excited for you all. Oh, sorry, my cat is not here. We're very excited for you all to hear these presentations from our wonderful academic experts. This collaboration came about because the Federation of American Scientists, we are a science and technology policy think tank that works to promote science as a solution to global challenges and make recommendations to the government on how to do that. And this project came about because we had been working on focused research organizations and trying to promote this new way of organizing research in order to tackle challenges that may be difficult to do from a purely academic or a purely industry position. And so focused research organizations are nonprofit research organizations designed similar to a startup, but with a very well-defined technical goal and like time-limited, budget-limited scope. And we are very excited to partner with Metaculus on this project since it's an opportunity to see how forecasting might allow us to better understand the potential rewards of these focused research organizations and also evaluate and get public input and expert input on the technical feasibility, technical impact that it might have on science. And so we're very excited for you all to hear more from the actual proposal authors themselves and get insight from their deep expertise. And I'll turn it back to Christian.
Thank you, Alice. And one thing I'll mention before I introduce Dr. Ethan Beer, our first speaker, is there is a Q&A interface. You can add your own questions, you can upvote questions, you can comment on questions, and during the Q&A portion following each speaker's talk, we'll have an opportunity to pose these to the speaker. All right, without further ado, I'm going to introduce Dr. Ethan Beer, who is the Distinguished Professor in the Department of Cell and Developmental Biology at UC San Diego. And he is, apologies, I'm just, his lab focuses primarily on two research areas, developmental patterning and using Drosophila as a model to understand human disease processes. And he's going to be talking about a focused research organization to reduce antibiotic resistance in aquaculture. All right.
So folks, can you see my presentation here? Is that visible? Okay, yes. So first of all, I'd really like to thank Christian and Alice, who've been I've had the pleasure to work with here over the last few months, bringing these exciting ideas of focused research organizations into some kind of framework to be evaluated in a clear way. And I am a very enthusiastic supporter of this concept, and I find this to be a really important unmet need. So I'd like to tell you a little bit about the idea that we have, and it attacks a big question, I think, in food security, which is fish such as salmon are a primary affordable source of protein worldwide, and yet there are a lot of problems with sustainably producing this kind of protein. Wild populations of salmon and other fish are seriously threatened. Many populations of fish, they're down to less than 10% of their original numbers. It's a serious problem just fishing them straight from the ocean, given how many people are currently on the planet. Open ocean salmon enclosures are one solution that has been tried, but there's an awful lot of frustration with this that comes from two problems. One, that they're very densely packed, and you get sea lice that form in there. And then all the waste products that come from that get out into the surrounding environment, and it's very damaging. And I just read this piece by Graham Lawton from the New Scientist, who is a devastating critique of that. An alternative is to have land-based aquaculture, which in principle is sustainable. And in fact, Graham admits that that's true too. But there are big problems that or challenges that face that land-based aquaculture, which again has to do with having sustainable numbers of fish that are grown in clean water, where of course the big problem is even with water recycling processes, which are well developed, you still have problems with bacteria, contamination, and growth. And so that's one area where we think we have a technology that we've developed that could potentially really make that form of protein production more sustainable. So this gets into the nuts and bolts of the whole CRISPR revolution, and I don't want to focus too much on this technology. It's one that we've been very much involved in a practical way. But the bottom line is what you have here is a programmable DNA cutting enzyme called Cas9 that together with the guide RNA is capable of cutting DNA in the genome pretty much anywhere one wishes. And so my lab helped pioneer a process that we call active genetics, wherein a genetic element is inserted into the genome exactly at the site where the program nuclease is programmed to cut. The nuclease is programmed to cut. And the way that works is you put the Cas9 and guide RNA genes in between homology arms that match exactly the cut site in the genome, and then those proteins are produced. They cut the genome in that location, and then through homology-directed repair, a process that is well understood and is particularly effective in the germline, the gene cassette gets copied into the break in the genome. And once that happens and it's in the genome, again, the Cas9 and guide RNA can be produced from that location. They cut the other chromosome. So if they're on mom's chromosome, they cut dad's chromosome. And then again, there's homology now along the entire chromosome, and the break is repaired by copying in the genetic information. And so now what happens is that both mom and dad's chromosome carry the genetic information. And so instead of it being transmitted only to 50% of the progeny, it's transmitted to nearly 100% of the progeny. And in fact, in the laboratory, in both fruit flies, which I study very intensively, and in mosquitoes, which we also study with collaborators as well, we can get it up to 99% of the time this gets transmitted. It's very, very efficient. And so we call this active genetics, and we've been working with a team to use this technology in a way that's often referred to as gene drives, where you take the genetic material that you would like to disperse through a population and you put it in one of these active genetic elements, and then it gets copied through a population every time animals breed. And so just to give you a very quick sense of it, if you have Mendelian standard inheritance of a trait where, for example, the green mosquito, say, is carrying genes that immunize a population against malaria, then it dwindles as time goes on, as it mates with other insects, other mosquitoes that don't have that trait. Whereas, and so the trait can essentially disappear from a population unless you put it in very, very large numbers. Whereas with a gene drive, what happens is that every time the mosquito mates with a wild type mosquito, it converts the genetic material that was inherited from that wild type mosquito so that all the progeny get the trait. And so then after 10 generations, essentially the trait is now present through the entire population. And that's about 1 or 2 seasons for mosquitoes. And so if you hook that genetic element that copies itself that way to an element that immunizes mosquitoes against malaria, then in principle you have a means for combating that disease. And I am part of a larger group that is trying to actually do that. And the technology is very promising. The question is, of course, how one gets that approved. And that's an interesting and important point that's relevant to all of this type of activity, which is one has to have very important ethical and social understanding of the impacts of such technologies. So I'd like to tell you just quickly about proactive genetics, which is an application of this same concept to bacteria and the problem of antibiotic resistance. And so standard processes that people have tried for several years with some success once the CRISPR technology was developed was to target the cutting ability to cut genetic information that's carried on these bacterial factors that confer antibiotic resistance. Plasmids are these small molecules that are in many copies, sometimes 50 copies or more per cell, that carry these antibiotic resistance determinants, and they can be spread between cells, because when a bacterial cell dies, that DNA can get picked up by other cells. It's a nightmare. It just keeps spreading, and it gets into the hospitals. It goes through the environment. It goes into, and in this particular case, it's a big problem in aquaculture. And so what people have done in the past is they take the Cas9 and the guide RNA, the two components that make the cutting, and they produce them in a bacteria in such a way that they combine, and then they cut the plasmid at the site of the antibiotic resistance, and they destroy the plasmid. And so if the plasmid has two different antibiotic markers on it, it destroys the plasmid completely. And so that if you then plate those bacteria on either antibiotic, you don't get anything because the plasmid has been destroyed. But that process is not incredibly efficient. It sounds good, but it only reduces the prevalence of antibiotic resistance by 100-fold, which is okay, but not great when you're really trying to have a big impact. So what we thought of doing is instead is to put a genetic cassette together that has homology arms and the guide RNA that's doing the cutting in between them. And so that now what it does is that when you induce the production of Cas9 and the guide RNA, instead of destroying the plasmid, it simply edits it. It inserts into the gene that you are trying to disrupt a genetic cassette, the guide RNA itself, that then disrupts the activity of that particular antibiotic resistance. And in the experiments that we did there, we could show that we disrupted the antibiotic resistance of the targeted antibiotic resistance gene, but not the other one. And moreover, that its effect was now 100,000-fold. So that's a very respectable reduction. And so that's the technology, which we call proactive genetics, that depends on a self-amplifying system, because it turns out the guide RNA is a limiting entity. And so the more it copies itself into the target vector, the plasmid that is at high copy number, the more effective it becomes. So it's a positive feedback loop. And we want to harness that in terms of aquaculture. And so we have taken that genetic information, that device, that platform, and we've put it into a large single unitary genetic element that can be transmitted between bacteria by the process of conjugation. And so that allows it to spread. It's essentially mating in bacteria. And so the way that works is that you take the one strain of bacteria that carries the genetic element with all of the goodies in it, and that took some engineering to build that thing. That's up there in the upper left corner. It took us over 1.5 years to do that from the first set of experiments. But then anyway, you put it into into test tubes with a test tube with the target bacteria, the strain that would be the problem, say, in the fish tank, and there you make use of a fact that it's a far more efficient process if you put those bacteria together with glass beads, because then the bacteria grow on the beads and they form a biofilm. And when they form a biofilm, this conjugation process is orders of magnitude more efficient. And so then if you do that, you can now go ahead and go through the whole process and reduce antibiotic resistance again about 100,000-fold, but by transferring it from one group of bacteria to another. So that's really, I think, a pretty impactful potential technology. So what's our idea? Our idea is to develop in the first 2 years a safe, proactive genetic conjugal bacterial strains that can deliver this, and also bacteriophage, because viruses, it's another delivery system that we're working on in parallel. Show that the Pro-AG strain can spread the antibiotic-resistant cassette in small fish tanks. That's the next step, which would be just to show that this technology is essentially working in a tank kind of situation. And then generalize that to a more significant aquaculture enclosure, which has got all the problems that you'd expect in it, like valves and so forth that biofilms could form on, where again, this proactive genetics would be very good at getting at those problematic aggregates of bacteria that would be difficult to get rid of otherwise. And then demonstrate in that for at least one antibiotic and for one bacterial strain that we can achieve 90% reduction of that in those tanks, so that basically now the people don't have to add large amounts of antibiotics to the water when they're doing the aquafarming, which is of course a big concern for people who want to eat those fish. So our team consists of myself as the project head, Victor Nizet, who's a microbiologist and antibiotic resistance expert, a colleague I've been working with for many years, a brilliant fellow. Justin Meyer, a younger person, an up and coming in microbial evolution, and he's a specialist in bacterial phage and how these things evolve. And of course, that's one of the big challenges, is you expect evolution to take place in these conditions. And so you want to understand how to do that. One of the things he's shown is that a situation where you have both a conjugal plasmid vector, like we've already built, and a phage vector, which we're working on, and you put them together, they end up acting together in a combinatorial way that is much more effective than either alone. And so we're looking at that synergy as another element. So the action team is a biology team, which would be people who come from our 3 laboratories. And one of them, actually from Victor's lab, already has an aquaculture facility that he's working in. So that would be a potential collaborator. An engineering team, the people who really understand the nuts and bolts of how you try to purify the water, reclaim it, and so forth. And this would also be generalizable to other things like, sewage treatment plants and so forth. This technology has got broader applications just than to aquaculture. Any environmental contamination in principle could be taken care of this way, particularly if it's in a confined place like a sewage treatment plant or an aquaculture farm. And then a bioethics team, that's critical because we really understand from all of our work with mosquitoes and malaria that you have to have a serious team of people who are looking at the ethical and social impacts of using any new type of technology like this one. And we have many colleagues that we work with based on our mosquito work, and they again would be very interested in working with us in this capacity as well. So I'd be happy to take any questions. Thank you for your attention.
Dr. Beer, thank you so much. First, I do appreciate, I remember you saying that you do have another commitment, so I wanted to give you the opportunity, if you do need to leave to make that commitment, then you can certainly feel free.
I'm good though, thanks.
Okay. Yes, so one question was, what do you see as the biggest technical challenges, risks, uncertainties in this proposal if it is funded?
Well, one always worries whether the thing will work, right? And that's a really big problem with funding, like from the NIH, and why I think a FRO-type situation is so perfectly applied to this. This particular project, believe it or not, I've submitted it to the NIH 6 independent times, and it's come close to getting funded twice, but then they won't go for it. And the reason is in part because they think it's kind of too risky, and they don't really understand, how it exactly is going to be put together with a practical application. And I think the problem with that is you can't do that for a small grant. That's just not the scale of the problem. So you need this intermediate scale, biotech company size scale to actually try this out to see if it will work. So it's high risk. B, it requires a lot of resources to test it out. And I think what we've got here is a very compelling case that this technology could work. It could work in a sort of absolutely unmet need, an important unmet need, to take a technology that's on the verge of being useful, it already is useful, but on the verge of being sustainable to being fully sustainable. So we could have a huge impact, but you can't do it without the funds to test it. And the biggest risk, of course, is that it won't work. But I think it's something that deserves a good test, and I think it's very likely that we'll at least be able to show that it can be working in certain situations, and then hopefully we would learn from that also by the scale of the effort what we might have to do, like bringing in, say, phage that would then complement the activity of just the conjugal transfer elements, so that now we do, through our analysis, come up with a tool that will give us the performance that we need. But I think, that's the biggest challenge, is to make sure that it works, and that, the details of what you'd have in a real aquaculture tank with all the valves that are coming in, the water going in, the water going out, making sure that you've got a system where you've got a safe strain of bacteria that on their own aren't going to cause any troubles, but that can deliver it very efficiently, or phage that, of course, won't cause any trouble on their own, that you can use to deliver it, some combination of those. I think those are the challenges. It's a technically challenging thing to do, but doable, in my opinion.
Thank you, Doctor. So we have Now, we do have a number of speakers to get through, but I do want to pose another question from one of our attendees and one of our forecasters. And I want to assure that all the forecasters that I will share all of your questions with the researchers, even if we don't get to it during this series of talks today. This question comes from Dima. Dima asks, you say bacterial strains, what species specifically, what known aquaculture pathogens are amenable to that gene drive that you've demonstrated in E. coli?
Well, there are quite a few different pathogens that you can be concerned about. The species of most concern to aquaculture are Aeromonas, Vibrio, Streptococcus, Flavobacteria strains that, as well as in some cases, some coli. And so we would probably start off in just using a single target species of bacteria, and that would be certainly something that is a concern, which is if we can prove it, as we would hope that we can, for a particular strain of bacteria, and coli would be a pretty good one, although it's possible that another starting strain that we could use would be one of the strep species. But in any case, that it would be important to show that you could not only get it to work in that one species, but that you could import that technology into other bacterial strains as well. And one thing I can say in support of the general concept is that people have shown that the CRISPR technology as a whole does work quite well in different bacterial backgrounds. And so I do think it's the same kind of problem that we actually have with mosquitoes too, is we can generate a gene drive in, say, Anopheles gambiae, but will it work in Colucci, which is another strain that causes a lot of trouble? And so you have to sometimes restructure it for a different species. But the hope is that as long as you've got the concept right, that is a barrier. It's something you have to think about, but it's doable. And the other thing I'd like to point out, just if I can just have one extra point to make, is that what's really cool about this is that if you get a species that isn't, a new species that comes up, you can build a new thing for that new species. If you get resistance in a given species to your system, you can devise a new element that targets the evolved species, because that you have control over what you're targeting with the CRISPR system. And so I don't feel like we ever win in a situation like this, but you keep in the competition. And I think this is a kind of technology that's so readily updatable that you can actually do that in real time and keep a balance going on where you don't necessarily win, but you keep it down. And if you drop it by 90% and you only have to use 10% of the antibiotics that you had to use before, that's a win.
Thank you very much, Dr. Beer. Questions, again, questions through the interface, I will make sure they get to the researchers. They can answer during the talks over text using the QA interface or later on over email, but we'll facilitate all of that. Next, I do want to introduce Dr. Conrad Cording. Here, let me spotlight you, Dr. Cording. All right. Okay. Dr. Cording is a full professor at the University of Pennsylvania, where he solves data problems in neuroscience and biomedical research. His lab focuses on deep learning, data analysis, and neurotechnologies. He's going to be sharing more on his focused research organization proposal to measure complete neuronal input-output functions.
Wonderful. Thanks so much for having me there, and on to something completely different to the previous talk. I want to talk about a call to let's do the experiments needed to simulate the worm. It's collaborative work with Andrew Leifer at Boyden and others, and it has primarily a scaling problem. That's why I'm here. So let's briefly phrase what the problem is. Imagine you wanted to understand a microprocessor, no? And I use that instead of a brain because no one really quite understands how brains work. How would you understand how a microprocessor works? Well, you can say you connect a few wires to an oscilloscope, you turn on and off the power of the whole thing, you kind of see what happens, and you kind of try and come up with models for that. The alternative is you can say, what if we could go in there and kind of put in little perturbations, little voltage pulses at all places and measure it? measure the effect everywhere. It's a bit like figuring out what the netlist is. And alternatively, what if you could directly give me the netlist? What I'll be arguing here is that part one is like how neuroscience works at the moment, where we're basically trying to figure out how brains, the most complex system that we know, work based on kind of correlations of what's inside with it. And strategy two, which is the strategy where we do lots of voltage perturbations and we see what happens to basically produce a netlist and understand what's going on there, is what we are proposing. Now, where are we proposing that? We're not proposing to do the human brain. Someone in Europe proposed that. It didn't end well, but we're proposing to do that on C. elegans. Why C. elegans? Because as you will see later, it seems that it should be possible. And what is so wonderful about this? It's a place in neuroscience where we can understand exactly how we can fail. And let's briefly talk about what we know about C. elegans. So the first thing is like C. elegans is actually super interesting. Most things that you can do, it can do outside of talking. You know, like it finds food, it avoids predators, it finds partners, it lays eggs, kind of like it has a really rich repertoire of things. It's much smaller. Keep in mind that the neurons in your head have about 10,000 inputs. That thing has maybe 50 or in most cases 30 or less. We can with new technologies produced by members of our team, we can now record all the neurons at all points of time. And we can identify the same neurons across animals. So it's always the same 302 neurons. And it's highly conserved in the sense that it seems that the neurons more or less do the same thing from one worm to the next. Now, big difference, average input number of like order 30 versus 10,000 makes it much, much easier to understand what's going on. It's much slower, therefore like recording is much easier than in mammals. We have complete input-output access, and we can pull across animals, which is really useful. So here's the high-level plan. We can do imaging. Ed Boyden has recently pioneered microscopes that see the entire worm, and we can see every single nerve cell, and we can use techniques pioneered by the team to basically see the activity of every single neuron, ideally calcium concentration, but we can also go for voltage. We can also use light to stimulate those neurons using optogenetic. So the idea is we're going to do experiments on them. We're going to record from all neurons. We're going to perturb lots of subsets. Then we fit models to that, and then we'll be able to simulate the worm. And I will hold that simulating any nervous system is going to be a major game changer for neuroscience. So let's be clear what we have here is we can perturb every single neuron. In fact, we can perturb lots of chosen subsets of neurons. So we'll really have all the things needed to reverse engineer the system. It's in a way we can really produce the netlist, as is important here. We can then simulate systems like that. Here you see one of our power calculations. If we want to have good predictions of what's going on there, we need a lot of perturbations and a long time of recordings from that. So much, in fact, that we need order 10,000 neurons if we go through it in detail. We can do statistical power calculations. We assume neurons are nonlinear. We can realistically, of the time scale of an FRO, go through all combinations of two neurons to be stimulated. In fact, do many of the three-way stimulations. We assume that we need about 10,000 parameters to describe every neuron. We can talk about the details. Is it 1,000? Some people think it's going to be a little less. Some people think it's a little more. The important thing is that gives us like a scale. Now you can say we can stimulate random subsets or even targeted subsets of neurons in all of the worms. We can, through that stimulation, make it that things roughly have a wide noise spectrum. I can't go through the details of the power calculation, but it seems that we need about 1 million minutes, which is basically 2 years of 24/7 recording, which is starting to be possible. So we're building an amazing team of people working on the perturbation pipeline, working on microscopes, perturbation recording. We have strong preliminary data that this is all doable. We need to massively increase the throughput there. We're also producing annotated connectome. That's a way of basically directly getting at a net list of which molecules are where, so that we can directly get that structure from function. And we then have a data team that's working on biology aware large scale fitting. And of course, we have a team with a lot of advisors from the C. elegans field, where we have a lot of prior knowledge. So just briefly, structure to function is our path to scale up. Here we can do molecular annotated connectomes. The idea is we do expansion microscopy on the worm, and then we can go and label different molecules, and we can also do in situ RNA sequencing, getting a lot of data. Our working hypothesis is that if we have all that data, we can predict what a synapse and what a piece of the neuron will do based just on the molecules that are there and the structure that's there that we already know. We know from prior research that without the molecules, we can't do that well. And then through that, but if we want to scale up to larger systems, we need a place where we can calibrate all those techniques, and the one place to do that is C. elegans. Now, the big question that you'll ask me is, why is this not traditional academia? Our main problem here is scale up. No, it's not that there's a specific question that we're asking. What we're saying is, if we want a good model where we can model every single neuron in here, we need order 10,000 experiments. We also need the experiments at a new level of quality. It's very hard to get a lot of these experiments to all be at sufficient quality. That now leads to the problem that, as you know, scientists tend to fall in love with ideas. I certainly do. This traditional academic system maximizes for publications, but what we really need instead is maximizing quality, and it's a rather just like scale up. How do we get 10,000 worms to such setups? And there's like relatively few small questions to ask, but we need those to make our students and postdocs happy. I think there's a big promise to change how neuroscience works by going from a traditional correlational hypothesis testing way that has not worked all that well in the past to a system based on figuring out what the causal effects are. Being able to simulate a whole nervous system is a major move of neuroscience into a future way that is much more like data-based. In doing so, we can also check how we can get there, rough timeline. We need roughly a year to optimize the technology to be able to scale. We'd need year 2 to start producing large scale data. Year 3 and forward, we'd start fitting at scale, and ultimately in year 4, so we'd be able to really do meaningful structure to function, and we should be able to reach at the necessary number of 10,000 worms in 5 years. I should also mention failing can roughly happen in year 2, because what you do if you fit is you have the standard that we call them learning curves, where if you do machine learning and you get more and more data, you get better and better. And based on how these learning curves look like, in particular, if you plot them in a log-log plot, gives you an idea on kind of if you are moving in the right way towards the things that you want to have. Our team is relatively early. We're currently still looking for good people to join us. Above all, most people on my team come from an academic background. That's where I come from, but we are currently trying to kind of grow our network beyond that so that we can build a team that can execute at this scale-up process. And with that, thanks so much for listening.
Thank you, Doctor. We have a couple questions in the chat that I'll pose to you now. First, Dima says, 50 inputs, roughly 50 inputs per neuron, yes, but can you reliably discriminate between them according to the stimulus? That's his first question.
Absolutely. No, what you do in this kind of a setup, you see every single neuron. And if you see every single neuron, what you do is you target one of these neurons with a targeted beam of light, because you see all of them. The problem of forward optics is the same as the backwards optic problem. So if you can show me where it is, I can target the laser at it. So in that sense, yes, you can target all of them. I think there's a deeper question behind it. Can I know exactly where on the dendritic tree it is? And it turns out that there is a little bit of variability from animal to animal, but it's relatively small, and we recently analyzed that. But this animal to animal variability is a major worry that we have, and we have ideas of basically using domain adaptation ideas from deep learning to deal with that with that variability.
Another question here: Are you assuming that stimulation is an on-off switch, or rather a variable resistor kind of stimulus?
Okay, this is a wonderful question. And in a way, we are at the mercy of biology for that. So the way all that stimulation works is you take existing kinds of channels, and these existing channels you then couple to a mechanism that couples into the optics. There has been a lot of work characterizing that and people working on different ways of coupling into those channels. But the best approximation for that is, as far as I understand it, still that it's a resistor where the resistivity is basically regulated by the voltage input. And that is, of course, something to be mindful about. There are approaches that turn off neurons as well that we'll use to get into a broader range there. But yes, it will still require kind of having good models of what exactly the stimulation does to neurons.
I see another question here in the Q&A. How do you ascertain if the perturbations you induce in the activation of the circuit or connectome will keep the worm's behavior in context or completely decontextualize it, such that inferring anything would end up being meaningless?
Okay, so this is there's a very deep underlying question here, which is, you can say in a given behavior, the brain lives in some state. But what you want to do if you want to simulate brains is be able to predict how it works across a large range of different states. And so what you want to do there is you want to make it so that the space of places that your network visits is relatively large. And then you want to fit for that, because that's where you get your so-called out-of-domain generalization from. So in that sense, the question is, what happens if we do these big perturbations? No? And there's two things. There's something that it does to the immediate downstream neurons that might be perfectly fine, but it also might be, if we stimulate strongly, that we kick the worm from one behavior to the next. But this kicking the worm from one behavior to the next is part of what we need to find out. If we want to do anything meaningful, we need to kind of say how the decision-making of the worm, the large-scale decision-making, oh, I'm now switching from eating to maybe running away or something, how these kind of switches happen. And for that, it's important to have that. But I also want to mention, we can record from all the neurons in the worm and even stimulate a good number of them while it's doing a standard array of tests. And we're collaborating with Steve Lavelle in that area.
Fantastic. Well, thank you so much, Dr. Cording. And again, if there are additional questions, you can respond to them in the Q&A interface, or we'll get them to you over email later. All right, I would now like to introduce the final team. I'm going to spotlight them here. Just one second. Just bear with me. All right, so I think that there may be a limit to the number of spotlights, so I will just trust this team to share your screens, and I believe you should have the spotlighting ability. So let me introduce you, and then I'll allow you guys to take care of that aspect of it. So this team is comprised of Chetan Raina, who has a long history of finance, investing, and operations experience, and he's the CEO of Icarus, which is an open science organization that performs side-by-side comparisons of antibodies to a particular target protein and publishes the results for use by scientists globally. Icarus is a collaboration between several organizations. We're pleased to be hosting, in addition to Chetan, Dr. Alid Edwards, the founder and chief executive of the Structural Genomics Consortium, a research organization that pioneers open science, particularly as it applies to protein science, chemical biology, and drug discovery. Also joining us is Dr. Peter McPherson, a James McGill Professor of Neurology and Neurosurgery and Anatomy and Cell Biology at the Neuro, where he's a member of the Neurodegenerative Disease Research Group. And then we're also pleased to host Dr. Carl Laflamme, a team leader of the Antibody Characterization Group, also at the Neuro. And they will be presenting on their proposal for a focused research organization to characterize antibodies through open science.
Perfect. Thank you, Christian. And thank you, Alice and team, and all of you watching live and the recording. We're pleased to present to you our project, Antibody Characterization Through Open Science. Carl, next slide. So in general, an overview. Life sciences researchers who study cells, human proteins, human tissues, purchase antibodies as a basic tool. There are over 6 million of them on the market from over 200 suppliers. The total market is estimated to be about $4 billion. And the big problem that we're trying to solve is that roughly half do not work as advertised. And so that represents a direct wastage of $2 billion. We've begun our testing of these antibodies, and what we've found is that of the 80 tests that we've conducted, we find that an average of 12 publications per test refer to and use bad antibodies in their paper, meaning that some aspect of nearly 1,000 papers is completely incorrect. So this is the monetary problem. This is the scientific problem. So our proposal is to test antibodies for each of the 20,000 human proteins. Our focus will be on recombinant and monoclonal antibodies, which are renewable, meaning once we've identified a product that works, essentially that product will be available in perpetuity to the scientific community. To one significant figure, we believe we can test all or antibodies for all human proteins for $100 million US. We believe that will save $2 billion in direct costs saved, and that's direct only. That excludes indirect time wastage, discoveries, entire fields going down the wrong path from the use of bad antibodies. So just the direct costs alone would represent a 20 times return per year, a one-time expense of $100 million to save $2 billion per year direct, and orders of magnitude more in indirect costs. I'll hand it over to Carl to tell you more about how we're doing this.
Hi, thanks, Jason. So yes, I'm Carl. I'm leading the science effort at the Icarus Initiative at McGill in the lab of Peter McPherson. So antibodies are critical research reagents used widely in academic labs and in the industry. Reason is industry allows researchers to identify, quantify, or capture a single protein within a chaotic mixture of thousands of different proteins. The ideal antibody would detect only its intended target in gray here in a particular experimental setup and no other protein. However, antibodies in different situations can not only detect the intended target, but as well as other unwanted proteins, or even worse, could identify only unwanted proteins. So the use of non-specific antibodies by researchers lies at the heart of the actual reproducibility crisis observed in biomedical research. Antibody must therefore be properly validated to ensure that only high-quality antibodies are used by scientists, and ideally, that poorly performing antibodies are removed from the market. Good, but who should characterize the million of commercial antibodies and how? So for 30 years, no one has wanted to take responsibility for this. Granting agencies are asking that scientists use well-characterized reagents, but do not necessarily provide guidelines or funding. Antibody companies provide application recommendations based on their characterization data, but we and others have observed that a significant portion of antibodies failed in recommended application when we use a superior characterization technique. Individual laboratories report that cost and time are major obstacles to the proper characterization of all antibodies they use. So we've decided to address the issue by combining an open science business model derived from the Structural Genomics Consortium to a technology platform that takes advantage of knockout cell lines to properly validate antibodies for 3 major research applications, which are Western blot, immunoprecipitation, and immunofluorescence. So Icarus works in an open ecosystem with 12 industry partners which consists of antibody manufacturers and knockout cell line providers. So all of them here are antibody manufacturers, apart from Horizon Discovery, which markets knockout cell lines, and Abcam, which is an antibody manufacturer as well as a knockout cell line provider. Our industry partners donate reagents to us to test. From 2019 to now, we've been working closely with all of them to establish robust and trusted standard operating procedures. We have every 2 months or so advisory committees with senior representatives from all companies. We go through all the antibody characterization data that we generated. Once we reach our scientific bar, we consolidate all data into antibody characterization reports, one report per protein, which we then publish openly, first on an open repository called Zenodo. On Zenodo, we have our own community where you can access all 80 antibody characterization reports that we have generated, and we are progressively including more and more reports. We are converting Zenodo reports into bona fide published peer-reviewed articles on the F1000 research platform. And we use our RID, so unique identifiers, when we test any antibodies. And these antibodies are linked to the antibody registry, where we can also find our characterization data. Selection of protein targets come from funders. For example, Michael J. Fox has funded various Parkinson's disease targets. So once targets are funded, we inform our partners, which in turn provide us with in-kind reagents. It is important to take in consideration the renewability of antibodies for a global antibody characterization effort like ours. Although we characterize all available antibodies for certain proteins, we and the antibody manufacturers are still focusing on recombinant antibodies. For recombinant antibodies, the light chain and the heavy chain are sequenced and cloned into recombinant in expression plasmids, enabling the same antibody to be produced repeatedly with the same performance from batch to batch. Polyclonal antibodies are common products, but are not renewable. Serums collected from animals have limited antibodies quantities. Antibody performance can vary from an animal to another one. And monoclonal antibodies derived from hybridoma cultures can produce a long-lasting supply of antibodies, but might suffer from genetic drift. We still consider them as renewable products. Okay, we have described the antibody market, the antibody liability issue, the ICRIS platform, and the various types of antibodies, but let's dive into the data. So here's an example where we screen antibodies for Western blot, IP, and IF. The name of the protein is TMEM106B. This protein target was nominated actually by various foundations due to the fact that it was recently shown to be present in brain inclusion from patients suffering from different neurodegenerative diseases. So for this particular protein, we've received 6 antibodies from 5 different suppliers. The name, actually the catalog number of each antibody is indicated above each bar, each Western blot. When there's 2 stars, this means that this antibody is a recombinant antibody. When there's a single star, this means that this antibody is derived from an hybridoma. And when there's no star, it means these are polyclonal antibodies. So cell lines that were generated from wild type and TMEM knockout cell lines and ran side by side on gradient SDS page. We stain the membrane with Ponceau S to show that equal protein lysates from the wild type and the knockout cells were properly loaded. Western blots were performed with the indicated antibodies. What you see here is an antibody that is specific for Western blot. As you see, this antibody detects TMEM106B, and we know this is TMEM106B because the band disappears in the knockout. Interestingly, this antibody is not recommended for Western blot by the manufacturer. The second antibody here do not detect TMEM106B. It detects unwanted protein. However, this antibody is recommended for Western blot. This antibody performs well. This is the top-sided antibody for Western blot for TMEM106B. It might detect TMEM106B here. We might see a band that disappears in the knockout. However, this antibody detects loss of unwanted proteins. I wouldn't use this antibody for Western blot. This antibody is recommended for Western blot. However, you can tell it's very dirty. And this antibody do recognize TMEM106B by Western blot. However, it's not recommended by the manufacturer. We then screen all the antibodies by immunoprecipitation. So we evaluate whether the antibodies can capture TMEM106B by performing immunoprecipitate in native conditions, followed by Western blot to evaluate whether the antibodies could enrich TMEM106B in the immunoprecipitate compared to the starting material, SM here. So this antibody is a great example. This antibody enriched TMEM106B in the immunoprecipitate compared to the starting material. And you can see that the unbound fraction, UB, the unbound fraction is completely depleted from TMEM106B. While most antibodies is able to capture this protein by IP, most of them were actually not recommended for this application. So our platform is also able to indicate new applications for certain antibodies. And lastly, we screen all the antibodies by immunofluorescence. We use a trick here where we label the wild type cells with a fluorescent dye. We label the knockout cell with a different fluorescent dye. We plate them together as a mosaic. This antibody here and our analysis code is able to differentiate between the wild type and knockout cells and segmentate the wild type cells in green and the knockout cells in pink. This antibody here is recommended for immunofluorescence. However, as you can see, the signal is exactly the same between the wild type and the knockout cells. This antibody here is also recommended for this application. However, the signal is very similar between wild type and knockout cells. Only this antibody is actually selective for TMEM106B by immunofluorescence. You can tell that wild type cells here have a clear signal, and that signal disappears in the knockout cells. In the knockout cell, the signal is pretty similar to the background. So a bit of history about ICRIS. So in 2018, it literally took me 1 year to characterize antibodies for one protein. Next, between 2019 and 2022, we grew our team to 3 scientists. We developed the protocols in collaboration with our industry partners. And we could do 10 targets a year. And the cost of it per target is actually pretty high. The current situation now is we're able to do 100 targets per year. The cost per target went down to $15,000. We were able to raise over $6 million of research funding. We have a team of 10 scientists, and each scientist can do 10 targets per year. We published in 2021 in New Biotechnology an opinion paper where we described what would be the cost of characterizing every antibodies for all human proteins using our approach. And as Chetan mentioned, it would be roughly between 50 million and 100 million. With 100 bench scientists, we believe we can characterize antibodies for 2,500 proteins. So after 4 years, that would be 10,000 proteins. So we believe this is a picture that we can follow. So in summary, we follow strict open science principles. We publish the data openly and as quickly as we can. We have the trust of leading antibody manufacturers. We offer something that is pretty unique, where we compare all commercial antibodies against single proteins, and we offer the data openly. Our platform is transparent and scalable, and we believe we can enable rigorous and reproducible research by providing scientists with a guide of the most appropriate antibodies for their research needs. and we're ready to take any questions.
So I appreciate that the team has been very responsive to the questions in the chat, and so I see that a number of these questions have already been addressed. That's fantastic.
I'm not surprised.
Let's see. Okay, we have one here, which I believe Dr. McPherson was in the middle of answering. And maybe I'll pose it to the group in case there are other responses, but also the group is free to peruse those other questions that have already been answered in case folks have different responses than what's already been given. But I see this one question. So why does industry collaborate with you? What's the benefit for industry to join into your ecosystem? And I can appreciate there's already a question to that, but it seems general enough. Perhaps there are more answers to that question.
Go ahead, Chetan.
Yeah, I was going to say Peter seems to have answered it, but it's really interesting from a business perspective. These are companies who are trying to do the right thing, and it is actually difficult to make good antibodies. And so these companies that we're working with use our test data to adjust their catalog, as Peter was saying in the chat. They highlight products that work really well. They use our data on their own websites as part of their data sheets. When an antibody performs very well, and when it performs poorly, what we found, and Carl and the team put out a review recently, that 1 in 5 of the antibodies that we have tested have been removed from the market entirely. And so these are good companies trying to do the right thing, and we're assisting that. We're helping them from a business, kind of strictly business perspective, market their good products, rationalize their catalog, decrease their support costs, decrease the phone calls coming in, the scientists saying, hey, my antibody didn't work, and then they have to say, did you leave it on the bench overnight? We're really helping them create efficiencies in their business from that perspective and put out better products for all scientists.
And it's important that when we do identify high quality antibodies, that the community knows about it. Not later than yesterday, our team, together with Thermo Fisher, did a webinar where we discussed the new antibodies for a certain protein called Parkin in Parkinson's disease. And now these antibodies, which are superb, are now well known by the community and can be used to promote better research.
Thank you very much. I have another question from the chat here, and then I think we will wrap things up. This question relates mostly to infrastructure. This is from Dima. What's the expectation of the number of laminar flow hoods and researchers/technicians working on the project?
Within the 100 bench scientists that would be required, we expect that 25 to 30 would be involved in cell culture. So I would suggest that, and if they use a cell hood half of their day, I would suggest 15 to 30 hoods would be required.
And Christian, maybe I'll jump in. I see what's the benefit of third-party characterization over creating a company that makes high-quality recombinant antibodies. Recombinants are great, but you'd still have to test them using knockout cell controls. So any product, however you make it, there are so many great methods, epitope, protein, you got to test it. And the proof is you got to put it to the test. So that's what we're doing. We take antibodies made from all different techniques and test them. So that's really the gold standard, in our opinion, way of making sure that that your product works.
Yeah, I was on the board of the NIH's antibody manufacturing thing, where every professor comes up with this idea: I can save the world and do it myself. And for the last 20 years, I've been part of 3 projects on the SABs of doing that. And it goes from 6 million available antibodies to 6 million and 150, because they make and then it becomes a cacophony of uncharacterized antibodies. So our opinion was, if we become the characterizer, then we could take advantage and leverage all the investment that's being made all over the world in making new antibodies and find the good ones. There's probably, for every company, they're investing hundreds of millions of dollars each. And so That I think it's a far more cost-effective way for society to characterize what's ongoing already than start yet another new antibody thing, that all of which have failed quite spectacularly every year. And what Jane does say, for every antibody, you need to make the antigen, which is the protein. The costs just mount up. And we tried that for 30 years and it didn't work, unfortunately. I tried, I failed spectacularly.
Yeah, and very quickly, I think, Carl, your statistics have shown that most 95% of human proteins already have, I think it's 2 or 3.
That's correct. That's correct.
So,
95% of, yeah.
You know, we think it's just, you know, it's cheaper and you have to test them anyway. So let's test what's out there instead of making more, as Al said.
We have one last question, but I understand also if any speakers need to leave, they have commitments. But I'll pose it to the group. And then this question is, how many new antibodies are coming to market every year?
Short answer is we don't really know. In fact, the 6 million that are on the market, we don't really know if that's a unique 6 million. There's a big problem with antibody duplication, relabeling, one company taking over another, one company sub-licensing another. And so that was a lot of words to say we don't know. It is a difficult market to analyze.
What's interesting is when we characterize all antibodies for a certain protein, and unfortunately no renewable antibodies are available or are specific for any application, we do revisit an antibody, a protein when there's new renewable antibodies marketed at a certain time point.
Yeah, and we have had a case where we show that the antibodies on the market don't perform well, and then the companies will go and take that as a business opportunity, go to their labs, create new products, and then we'll test those products. And then in that Parkin example was great, and then we can highlight that directly to the Michael J. Fox Foundation's researchers on Parkin. So some of what we are doing is actually like spurring the creation of new products and shaping the R&D to parts of the market where we don't actually have good renewable antibodies yet. Another reason folks participate with us, so that they can direct their R&D most effectively.